Okay, so in the last lecture, lecture 4, we talked about complementation analysis and how you determine whether two genes are in the two mutations are in the same gene or not, right? Now we will come to another type of analysis, which is referred to as epistasis analysis. Okay, epistasis means that if you are trying to say we're talking about a pathway, a biochemical pathway, a signal transduction pathway, something like that, where multiple genes act in a certain sequence. Okay. Now, from this mutational analysis, can you determine the sequence of action? And the answer is yes, and that is done by epistasis analysis, and it is done not only in yeast, but it is also done in flies and sometimes even in mouse. Okay, so the first one, okay, it's a rather straightforward one. It's a linear pathway. One is substrate. Let us say that this substrate is blue in color. 2 is the product of that substrate because of the action of enzyme A. Let us say it is green in color. And 3 is the product of action of enzyme B on the green colored product, and the product is red in color. These are made up, okay? Don't take it too seriously. These are made up, but it will help us understand the concept. Now, if either A is mutated or B is mutated, R, the red colored product, will not be formed. There is no question about it. However, you look at it, if A is mutated, then the substrate B itself will not be consumed. So B will be, the blue colored substrate, substrate 1 will be accumulating in the A mutation. And in if gene B is mutated, then the substrate 1 will be converted to product 2, which is green in color, but that will not be consumed. So the green colored product will accumulate. Now, if both A and B are mutated, then what will happen? It doesn't matter that B is also mutated, because A is gone, meaning substrate 1 cannot be consumed. So the blue colored product will be accumulating. So you can tell because of this that A is upstream of B, that A acts first, then B acts. If it were just the other way around, if B were acting upstream, like B were acting first and A were acting second, then the products that will be accumulating will be different. If B were acting upstream, then a mutation in B will have accumulation of the blue colored substrate, and then a mutation in A will have accumulation of green colored substrate. That's not the case. So this is how you can order the genes as per their sequence of action. That is one kind of explanation, one kind of use. There's another kind of use, which is if there is a signal transduction pathway. So let us go by the example that is given here, that you have a reporter gene. A reporter gene is what? A reporter gene reports activity of other genes. Okay, let us keep it simple at this point. We will elaborate on it in a later lecture. So what will be a typical reporter gene? Many of you may have heard of this gene called green fluorescent protein, GFP. Okay, you engineer expression of GFP in an yeast. You know that normally GFP is expressed in jellyfish, okay, in the sea. So it is not a native gene for either yeast or mouse or Drosophila, none of this. So these are all engineered. Now, when you just put the GFP, the ORF, the start codon of GFP till the stop codon of GFP, no GFP can be produced because there is no mechanism for it to be transcribed. So you have to put the necessary DNA elements for it to be transcribed. And the moment you put the necessary DNA elements for transcription, there is an element called enhancer, which dictates the time, space, and quantity of the production of the gene. And in this case, it is GFP. Okay? Now, this is a system that is used very widely in molecular genetic analysis if we want to understand regulators of the DNA element. Okay? So if you knew somehow that this DNA element confers this kind of an expression pattern in a gene, by expression pattern I mean spatial pattern, temporal pattern, and quantitative pattern. Okay? And you want to know what are the genes that are involved in interacting with this DNA element that allows the reporter to be expressed in a particular manner. So then you can do a mutational analysis and do all sorts of things. Now let us say you have done such a mutational analysis and you have identified two mutants. One is A and the other is B. Your observation is that, do not look at this plus minus first. Okay, first look at the observation. Your observation is that a mutation in A means that GFP expression goes down. Let us say this reporter is GFP. Let us say this GFP. So if there is a mutation in A, GFP expression goes down. But if there is a mutation in B, you get a lot of GFP expressed and everywhere. In every cell GFP is expressed. That's what it means by constitutive reporter expression. Meaning the regulation that it had regarding space and time is gone. Now, interestingly, if there is a double mutation in both A and B, then you see that the expression is still suppressed. Okay, so think about it another time. Just mutation in A suppress the reporter expression. So you knew that A positively regulates the expression. Just mutation in B caused constitutive expression of the reporter. So you knew that B, when present, blocks the expression of reporter. These two you knew. What you did not know is the order of action. Because the double mutation reproduced the phenotype as the mutation in A, so A must be downstream. So irrespective of what, when it comes to the action of A, because A is not there, so if everything above is fine and all right, you will still not have the A function, and therefore the gene expression will remain suppressed. So that's how you knew that between the two, B and A, B is upstream, A is downstream. Because the other way around, if you look at that, I have drawn it here, that if B regulated it positively, like if B regulated A positively and A regulated the reporter negatively, what would be the phenotype in just mutation of A, just mutation of B, and double mutation? Let us look at it. So A negatively regulates the reporter, right? So normally if A is present, the reporter will not be expressed. So if A is mutated, then that negative regulation is gone. So you will have more than normal reporter expression. If B is mutated, B helps the production of A, right? So if B is mutated, then A will not be produced. So it is as good as A being not there. So that will also give you enhanced expression of the reporter. And the double mutation will also give you enhanced expression of the reporter. So that way you can order them. So these are, very interesting, like these are like puzzles, okay? You have these individual data, the mutant phenotype in A, mutant phenotype in B, mutant phenotype in AB double, and you have to order them. So you will try to fit the jigsaw puzzle, and you will get it. It is a lot of fun. Okay. Now, if it is other way around, that A is upstream and A, like, okay, forget about it. Let us look at the data again. That if A mutation represses the reported gene, B mutation represses the reported gene. So these two conditions remains the same for this and this. Okay, but the third condition, this one varies. That in this case, double mutation gave a reduced amount of reported expression. In this case, double mutation gives an enhanced amount of reported expression. So how do you do it? So this is when you can tell that B negatively regulates the reporter, because that is when, if you have a mutation in B, you will get constitutively active reported expression. And because A gives repressed reported expression, therefore A cannot positively regulate B, right? It cannot do that. Because if A were positively regulating B, I'm saying the hypothetical situation, that if A were positively regulating B, then what would have happened? If A is not there, then B expression would go down. And if B expression would go down, then there will be constitutive expression. So that is not the case. We know that the data says that mutation in A gives repressed reported expression. So therefore, A must be regulating B negatively. So why would mutation in A give repressed expression? Because A normally was not allowing B to come up, right? But if you mutate A, then that negative regulation on B is gone. So now B will be expressed. And if B is expressed, then it will suppress reported expression. So this way, it can work both ways. That if you have these observations, phenotype in each of the single mutants and in the double mutant, you may be asked to construct the pathway, or you have been given the pathway, you can be asked to predict what will be the phenotype in mutation of A and mutation of B and double mutation. Okay, so this is how, through epistasis analysis, you can order action of genes. I hope I could make it clear to you. Now, there is another kind of analysis that can be done. These are called, these are the kinds of analysis by which you get to understand or identify interacting proteins. Okay, so think about it. We are going back to our very popular favorite example of galactose metabolism pathway mutants. So you have 3 different mutants, A, B, and C. Mutant A, and you have determined through complementation analysis that these are all in different genes. They all belong to different complementation groups. Okay. Now, mutant A, you don't get growth in galactose. Mutant B, you do not get growth in galactose. But if you cross the haploid of mutant A and haploid of mutant B, suddenly you may get growth in galactose. Now, that is very interesting, right? That these are, so these are in the haploid, by the way. Okay, you have to be very careful here. These that are shown here, these are in the haploid. Okay, these are not diploid mutations. So that means that you will take mutant A, you will take mutant B, and then you will do tetra-dissection, like you will make the diploid, you allow it to sporulate, and you will get the individual spores, and you do tetra-dissection, and by some mechanism you figure out the haploid where both A and B are wild type, where only A is mutant, B is wild type, where only A is wild type, B is mutant, and where both are mutants. Okay, but these are all in haploid. Because in diploid, this does not even work. Like, it does not matter, because you will get suppression if the mutations are in two different genes. Now, how does it work? Okay, let us think what happens in mutation. Mutation is in the DNA, and we are, generally speaking, we are not talking about nonsense mutations, okay, or silent mutations. We are talking about missense mutations that change one amino acid or two sometimes, okay, and that gives you the phenotype. That if in the haploid just A is mutated, it will not grow in presence of glucose. If in the haploid just B is mutated, it will not grow in presence of glucose. Now, why? Now let us take a very simple example. Let us imagine that both A and B are two enzymes. Okay? And they are two subunits of an enzyme, let's say. Let's say two subunits of an enzyme. I am giving you a very simplified example. It doesn't mean that it always acts that way. Let us say that it is the two subunits of an enzyme. So what does a mutation do? What mutation does is that it slightly changes the shape of the protein. And that is denoted here in your textbook cartoon, that instead of this little protrusion that is like a square, in the mutant it is like a triangle. Okay, and the wild type B, which has a little indentation which is rectangular shaped, in the mutant it is like an indentation, but it is triangular shaped. So in either of these cases, the interaction of A and B, which is very nice here, is not possible. So if they cannot interact properly, they cannot form the functional enzyme. So like you say, the mutant phenotype. But if by chance it happens that the mutation in B creates a change in the shape that is just good enough to accommodate the changed structure of A product after mutation, so that the interaction is restored again. Now this you do not do in a directed manner. Okay, you do not say, like, you do not set out to create a mutation in B that will be just good enough to accommodate the mutation in A. So the way it is done is I'll now tell you how it is done. Okay, I first explained what it means, but now I'll tell you how it is done. Let us say that you got this mutant A, okay, and you figured out that it is an enzyme, okay. Maybe you raised an antibody against it, maybe you raised an antibody against it, and you biochemically purified the protein, okay, and you saw that this A containing protein complex has a higher molecular weight than the molecular weight of A. Okay, so you knew that this A is interacting with some other proteins. Okay, so you want to know what those proteins are. You can of course do biochemical ways, but the way these genetic experiments evolved in older days, when getting from the mutant to gene to protein was not that simple. So what scientists should do, they will use more genetic tools. So how would they do it? So you have the mutation in A, Then you take that as the starting strain. It's a haploid with mutation in A. And then you do a fresh round of mutagenesis on that haploid A, mutated haploid A, and look for any haploid yeast that will be able to grow in the presence of galactose. Please note that mutant A by itself is incapable of growing in the presence of galactose. But now you are trying to see that I start with a strain that cannot grow in presence of galactose, and I mutate it randomly using the standard mutagens. And do I get any mutants that will now regain the ability to grow in presence of galactose? These are, like, if you just use plain English, this is reversal of the phenotype. The phenotype was inability to grow in presence of galactose, so that is reverted. So these are called revertants. But revertant in genetics generally means something very specific. That is, the mutation that was responsible for A's loss of function, that specific mutation changed and brought it back to the original sequence. That is generally what is meant as revertants. The rest are called suppressors. Okay, that is, another mutation happened in another gene which suppressed the phenotype. How would you know that? How would you know whether it's a suppressor? That by whenever you are thinking of suppression, by definition, at least two genes are mutated in the cell, right? So if you take this cell, this haploid, and cross with this haploid, okay, which is a wild type, then in the diploid you will have the good copy of A, a good copy of A, and a bad copy of A, and a bad copy of B, and a good copy of B. Now if you sporulate it, then you will get spores like 2 out of 4, statistically speaking, will not have the ability to grow in presence of galactose. Okay, so you know that this is a suppressor. This is not a revertant. Okay, so this is, when one mutation, when when you created one mutation in the backdrop of another mutation, the resultant is actually wild type behavior. Okay, it is not wild type strain, but it is wild type behavior because it can grow in presence of galactose. But the reverse can also happen. That, say, mutation in A is not like, they do not grow in presence of galactose, in absence of galactose very happily, but they can grow, like limping, crawling, not running, but it's growing. Similarly, mutation of B, it is not completely debilitating, that it did not arrest completely growth in absence of galactose, in presence of galactose as the only sucrose source, the only carbon source, but it was not a very happy situation. Now, okay, yeah, sorry. So what I was saying is that independently, either mutation in A or mutation in B, it is not that they just cannot grow in presence of galactose as the only sucrose source. They can grow, but not as well as the wild type. But when you combine the two, A and B together, then you see that there is absolutely no growth. So what it means in molecular terms is that A and B are also interacting partners, the mutation in A was such that it weakened the interaction with B, not eliminated. Mutation in B was such, like in this case, is that it weakened the interaction with A, but did not eliminate. But when both A and B are mutated, then they just cannot interact, just falls flat. So that is an example of synthetic lethality. Why is it called, like, like if you emphasize on the English terms, you actually get a whole lot. Synthetic, meaning because of synthesis of two, okay? Individually, either A or B was not lethal mutations. But combining A and B mutation causes lethality. So this is synthetic lethality. Okay? Synthetic lethality can happen in one of two cases. One, when the two proteins are directly interacting with each other. Okay, and so that is a good way of finding interacting partners. So if you are tasked with finding interacting partners, say like my favorite example is this, that my favorite way of asking questions is like this. Let us imagine that you discovered a completely new organism which is exactly like yeast. Okay, can grow in haploid, it can grow as diploid and everything, and it grows in some strange location. So now you are trying to understand the molecular biology in that yeast. The reason I create this kind of a background is that you cannot hide behind the fact that the genome sequence is known, will simply answer everything by genome sequence. I really want you to use the concepts of yeast genetics. Okay, so that's how I set it up. So if you have such an organism and you have conducted a mutagenesis screen, say let us give an example that, say you found yeast that grows in Yellowstone National Park, hot water springs. Okay, there you have very high concentration of sulfur, H2S, high temperature, and so on and so forth. So you replicate that kind of a condition on a Petri plate, and the yeast grows there happily. But unlike your normal yeast, which grows generally at 23, 24 degrees centigrade, this one grows only at 65 degrees centigrade, because Yellowstone's temperature is that high. Now you want to know which are the genes that makes it capable of growing in that high temperature, or which are the genes that are responsible for its ability to grow in presence of H2S. Those kind of questions, you can then do a screen, and you will get some mutants and indication of some genes by your first screen. Then you want to know what are the interacting partners, and then you can do a suppressor screen or a synthetic lethality screen. Okay? So you just have to design the screen a little bit smartly, and you will get all of that. But synthetic lethality can also happen due to another reason. That is, there is a product, whatever that product is. Now this product can be formed by two independent pathways. Okay, pathway A and pathway B. Pathway A is regulated by gene A, and pathway B is regulated by gene B. So if only A is mutant, then you will still make the product, but a little bit of it. So it will not be as good as the wild type, but it is almost wild type. If only B is mutated, you will also have a reasonably happy animal. But if you combine the two mutations and you have both A and B mutated, then you will have absolutely no growth. Okay? So that is also synthetic lethality. So the difference between this and this is that here by synthetic lethality you get interacting partners, and here the synthetic lethality means two parallel pathways. Now, after you have gotten a pair of synthetic lethal alleles, you will then have to do further experiments to figure out whether we are talking about two interacting gene products or two gene products that are acting in parallel pathways. Okay. And the last point that I would like to talk to you about is that, again, we need to think about the day and time when, genome sequencing was nowhere, even on the horizon in the 1970s, even mid 80s, when I started my grad school in 1995, genome sequencing effort kind of started at around that time. And, I will tell you those stories when I come to those chapters. Okay. But, in the olden days, genome sequencing was not an option. But you still want to know whether, as much as about the organization of genes and chromosomes, etc., etc. So one of the ways of doing that is called linkage analysis. Okay. So linkage, physically, it means that genes that are present on the same chromosome and how close they are on the chromosome. The way it is done is you have to deploy, like you have a mutation in B gene, you have a mutation in A gene, and you have deployed by crossing a MAT A and a MAT alpha of B mutation and crossing a MAT A and MAT alpha of an A mutation, and you created these diploids. Okay, these are wild type for A, homozygous, but mutant for B, homozygous. This is wild type for B, homozygous, and mutant for A, homozygous. And now this is the, just for the sake of it, I'll tell you that A and B in this particular example are on the same chromosome. Because if they are on different chromosomes, there is no linkage, so it does not matter. They are on the same chromosome. So you are looking at in the diploid that came out of this cross, and there you can see that one chromosome will have, because the chromosome that will come from this parent, it will obviously have wild type A and mutant B, and the chromosome that will come from this parent will obviously have mutant A and wild type B, right? So this is a diploid. Now this diploid, which is called a double heterozygote because it is heterozygous for A and heterozygous for B, you cross it with what is known as a tester strain. A tester strain will have like homozygous mutation for both the alleles, A and B. Okay? Now you cross them and you see that how many will come up with genotype very similar to this. Okay, so that will be, sorry, I take that back. I take that back. Okay, so the genotype will have, see the way it is done, because it is a double homozygote for both A and B. So the chromosome that you are getting from this parent is the same. Okay, it does not vary, right? In all the cases, it is the same. Okay, but the one that you get from here, normally you should get AB like this, or you should get AB like this, but you may get some from this one, which is wild type for A and wild type for B, or wild type for A and wild type for, mutant for A and mutant for B. So these are not the parental type. Okay, in the parent, neither of these existed in this parent. Okay, in this parent, it was always, if it is like the two chromosomes the two haploid set that you get, it will always have wild type A mutant B or mutant A wild type B. So these are the parental types. These are new types, which are referred to as recombinant type. So this recombinant type can only be explained here, that during metaphase 1 of meiosis 1, when there is when there is crossing over of the chromosome, the father's chromosome and the mother's chromosome and genetic material is exchanged, if the two genes are very close to each other, then chances of a crossing over happening, so this is pure probability, right? If two genes are very close to each other, then a chance of a crossing over happening in between these two loci is low. But if two genes are very far apart from each other, chance of a crossing over between these two loci is relatively more. So by looking at the number of recombinant gametes over the total gametes, you can estimate what is the genetic distance. How do you do that? You take certain gene pairs for which you know the distance, okay, and you see what is the recombination frequency there. You know, it's pretty much like any titration graph that you do. So you have a physical distance and recombinance frequency table mapped, and then for two unknown loci, you look at the recombination frequency. Then you will be able to map the genetic distance. This genetic distance, the unit is called centimorgan. Centimorgan is in the name of Thomas Hunt Morgan, who is considered the pioneer in Drosophila genetics or fly genetics. But the unit was defined by his undergraduate student, Sturtevant. I cannot pronounce his name right. So in respect of his reverence to his mentor, Sturtevant, when he standardized this recombination frequency based genetic distancing method, he named the unit in honor of his mentor, Thomas Hunt Morgan, and it's called centimorgan. Okay, so this is used in all sorts of organisms. Okay, so that is pretty much it. I will stop today, and we will meet again for lecture 6. Okay.